Hi everyone! This video is about phenotype shifts, or rather, how selective processes, such as natural selection, artificial selection, or sexual selection, can affect the distributions of phenotypes in a population. In class, we've been looking at changes in allele frequencies, so now we're going to scale that up to see how those changes can affect actual phenotypes. Let's start by looking at a typical distribution of phenotypes for any given trait in a population. In class, we've mostly been looking at simplified examples of traits that have only two possible phenotypes, such as the hippos that can be either green or blue. But in nature, you know that's not really the case. For example, you know that not all humans have either just blonde or just black hair. Rather, there's a whole range of phenotypes for hair color. And this is what we find for many other traits as well. So in reality, phenotypes often exhibit a bell-shaped distribution curve, something that looks like this, where if our x-axis is the range of possible phenotypes and the y-axis is the number of individuals who have each phenotype, most of the individuals are somewhere in the middle. So many individuals have the intermediate phenotype, and then fewer individuals have the really extreme phenotypes over on the ends here. And a classic example of this is human height. So if you think of all the humans on the planet, it's really common for people to be between about 5 and 6 feet tall, with the majority of people falling somewhere in the middle, but less common for humans when they're adults to be 4 feet or under, or 7 feet or above. So this is another example of a trait in which we would see this bell-shaped distribution curve. So how do these phenotype distributions, or these bell-shaped distributions, shift when populations evolve? There are three common patterns that we see in nature. They are directional selection, stabilizing selection, and disruptive selection. And these drawings down here are indicating what's happening to the bell-shaped distribution curve, where the red is showing the initial and the blue is showing what would happen after the selective process. So in this video, we're going to go through each one of these in just a little bit more detail. So we'll start with directional selection. So this is what happens when there is selection for the phenotypes at one extreme of the distribution and against the phenotypes at the opposite extreme of that bell-shaped curve. So for an example, here I'll show you a population of lizards. Now these are not all the lizards, I couldn't fit all of them on this page, but you can see that these lizards have some differences in their phenotypes. Notably, they have different tail lengths. So some of them have really short tails, some of them have medium tails, and some of them have really long tails. And I'll just tell you that in this population, more of the individuals have the medium length tails. But there's also something kind of interesting going on, and that has to do with what the female lizards prefer. They're okay with the short tails. They like the medium length tails, but the lizard ladies go nuts for those really long tails. So if we were to track this population for several generations, we might see a shift in the typical tail length. So if the blue is our original bell-shaped distribution, and we're looking at tail length on our x-axis and how many individuals have each tail length, we might see a shift in this direction after the selective process. So our phenotype distribution would shift in one direction. So that's directional selection. The next phenotype shift is stabilizing selection. And this occurs when there's selection for the intermediate phenotypes in the distribution and selection against the extremes. So for this one, I'll show you an example of a population of mice. And you can see that there's variation between individuals. They range from very dark to very light mice, and they live in an environment where their background is mostly sort of a medium gray. Now, if they have a visual predator come into their environment, the really dark mice and the really light mice are not going to do as well because they really stand out against their environment. And so if we were to track this population over many generations, we would probably see that a few years later, most of the mice were sort of a medium gray because the alleles coding for a very dark coat or a very light coat didn't really get passed on. Those mice didn't survive. So if we graph this out with fur color on the x-axis and the number of mice with each fur color on the y-axis, if our original population had this kind of bell-shaped distribution, at the end of the time that we were watching the mice, we might see something more like this, where the intermediate is doing better. So the phenotype distribution becomes narrower, fewer individuals with the extremes, and then the intermediate phenotypes become more common. The last type of selection you need to know is disruptive selection. This is also known as diversifying selection. And this is when there's selection for the extreme phenotypes out at the edges of the bell-shaped curve and selection against the intermediate phenotypes. So for this one, I'll give you an example of a population of fish. And you can see these fish are all different sizes, ranging from very small to quite large. And they have a bell-shaped distribution curve. You can't see all of them here, but take my word for it. Now, if a predator comes into their environment, they're going to have a slightly different situation. And in this example, this predator can't really eat these big fish. They're too big for it. And the predator's not really interested in the small fish. They're too tiny. They just swim away. There's not a lot of meat on them. It's not worth it. And so this predator is mostly going to eat the medium-sized fish. So if we were to watch this population for several generations, we might see that the big fish do really well and reproduce, and the very small fish do really well and reproduce, but not so much the medium-sized fish. So if we were to graph it out, if the blue is our initial distribution of fish sizes, we might see that over time, the really small fish increase and the really large fish increase. So our phenotype distribution would actually bifurcate. It would fork into two sides like that. And we would see that the extreme phenotypes become more common and the intermediate phenotypes become less common. And something that's interesting to think about here is if this goes on for long enough, are these two groups, the really small fish and the really large fish, going to become different enough from each other so that we would consider them two different species? That's something we're going to talk about in class very soon. But for now, those are the 3 types of phenotype shifts you need to know. So please make sure that you can identify and describe each one. Until next time, take care of yourself, take care of each other.